Very good morning to our lovely lecturer, Madam Munira, and my fellow friends. My name is SALWANA ARINA binti AKHIR. My metric number 23 DBT 20F2005. Today I'm going to talk about DNA interactions. For information, DNA interaction is a method to purify DNA by using physical and or chemical method from a sample, separating DNA from cell membranes, protein, and other cellulose components. Okay, let's see what is the use of DNA interaction. For information, the use of DNA interaction for some medical conditions, DNA interaction is necessary to officially diagnose it, especially if the medical condition is genetic. Common examples include cystic fibrosis, Huntington's disease, or Down syndrome. In this experiment, you need to isolate some DNA from a human test subject. Why, you may ask, do you need human DNA? Okay, let's see. DNA interaction is typically the first step in a longer laboratory process. DNA interaction is an important part of that process because the DNA first needs to be purified away from protein and other cellulose contaminants. The skin on the inside of our mouth loses thousands of cells every day. These are the cells we are after. Okay, next, let's get started. The step you will follow to purify DNA from a sheet swab are shown below. First, collect sheet cell. Second, brush shell open the relief DNA. Third, separates DNA from protein and debris. Lastly, isolate concentrate DNA. Here are the material and equipment you will need to purify DNA from sheet cell. Drag the swab over to the inside of the test subject's mouth to collect a sample. Next, you place the swab into an expander tube. Look closely at the swab. Note that it's covered with hundreds of tiny sheet cells. Inside each sheet cell is a nucleus, and inside the nucleus is DNA. Next, using the micropipette, add some lysis solution to the tube. Lysis, a Greek word that means to separate. Then, place the tube into the warm water bath. Next, the lysis solution you just added contains two important ingredients, detergent and enzyme called proteinase K. The cell have stay in the warm water which long enough for the DNA to be freed from the cell, and we have removed the sweat from the tube. Next, add some concentrated cell solution to your tube. The salts causes protein and other cellulose debris to clump together. Then place the tube into the centrifuge. Inside the centrifuge, the tube spins around at the high speed. The heaving clumps of protein and cellulose debris to sink to the bottoms of the tube, whilst the strands of DNA remains distributed through the liquid. Next, use the micropipette to carefully remove the top liquids and place it into a clean tube. The proteins and other cellulose debris stay behind. Then, add some isopropyl alcohol to the tube. Inverting the tube several times mixes the isopropyl alcohol into your DNA solution. Because DNA is not soluble in isopropyl alcohol, it comes out of solution. Next, place the tube into the centrifuge and clip on the lid to close it and turn it on. Lastly, once the liquid is removed and the DNA is allowed to dry, you can redissolve it in the solution of your choice. You can store it in the freezer for many years, or you can move on to your next experiment. You have just extracted DNA. That's all for me. Thank you for watching.